Are you falling behind on crucial breakthroughs in antimicrobial research? The fight against multidrug-resistant pathogens like Acinetobacter baumannii is intensifying. Today, we're diving deep into a groundbreaking research frontier, serum-stable peptide prodrugs. This innovative approach could redefine our understanding of targeted bacterial studies. Stay tuned to discover how these precise mechanisms are being explored.
Acinetobacter baumannii is a highly dangerous multi-drug-resistant pathogen facing modern research systems. This opportunistic bacterium thrives in lab environments, affecting various research models significantly. It rapidly acquires resistance to multiple research compound classes, a serious challenge. Consequently, researchers often face limited or ineffective compound options for study. WHO classifies Acinetobacter baumannii as critical, emphasizing urgent need for new research strategies. Researchers explore innovative approaches, moving beyond traditional broad-spectrum research compounds. One promising preclinical strategy involves serum-stable peptide prodrugs for targeted investigation. These prodrugs activate only inside Acinetobacter baumannii cells for highly specific studies. Though early, this targeted approach could redefine how resistant infections are researched. Acinetobacter baumannii is notoriously difficult to effectively study and manage in labs. Its reputation stems from biological resilience and environmental adaptability within research settings. It survives on surfaces and tolerates harsh lab conditions for prolonged periods. The bacterium rapidly evolves under research compound pressure, a constant challenge. Carbapenem-resistant Acinetobacter baumannii is common in intensive research units globally. Traditional compounds struggle due to Acinetobacter baumannii is robust outer membrane. It also produces enzymes that deactivate research compounds before reaching targets. Efflux pumps actively remove research compounds from the bacterial cell, complicating studies.
That's the challenge. But what about the innovative solutions being explored? We're about to explore the precise mechanisms of how these peptide prodrugs operate. Before we dive deeper, remember to follow peptides.today for all your essential peptide news and insights, and connect with me, Sonia Rao, for detailed pipeline analyses. Now let's explore this targeted strategy.
Instead of overpowering with high doses, researchers focus on precision in their studies. Serum-stable peptide prodrugs represent a fundamentally different research strategy for targeting. Compounds remain inactive in serum, activating only after entering specific bacterial cells. The concept involves linking an antimicrobial to a peptide cleaved by specific Acinetobacter proteases. This releases the active compound exactly where needed, minimizing off-target research effects. This approach limits systemic toxicity in models and spares beneficial host microbiota. Success relies on peptide linker serum stability and efficient cleavage by bacterial enzymes. Substrate phage display identified promising peptide linkers from hundreds of candidates. Preclinical testing showed strong bacterial specificity and improved serum stability for these peptides. Inside Acinetobacter baumannii's periplasmic space, the peptide linker is cleaved selectively. This releases the active compound, increasing intracellular concentration and antibacterial potency in research. Existing research compounds for Acinetobacter baumannii use more traditional mechanisms. Cefiderocol uses a siderophore strategy for entry, then functions as a conventional beta-lactam. Sulbactam-durlobactam combines a beta-lactam with an inhibitor to counter enzymatic degradation. These current therapies lack the same intracellular targeting level as protease-activated prodrugs. Peptide prodrugs control activation, potentially improving efficacy while lowering systemic exposure in models. Serum-stable peptide prodrugs targeting Acinetobacter baumannii are currently in the preclinical stage.
The rise of multidrug-resistant Acinetobacter baumannii demands innovative research. Serum-stable peptide prodrugs offer a promising alternative, aligning compound activation with bacterial biology. This strategy emphasizes precision and targeted delivery. Continued preclinical validation is crucial for advancing this approach. Follow me, Sonia Rao, and Peptides.today to stay informed on this vital research pipeline. Before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment.